So welcome back to Biomindscape. Now we are continuing the chapter of molecular basis of inheritance. In this chapter, we will know about the transcription and the remaining part. So in the previous video, we discussed about various experiments and what is inheritance, DNA, the RNA world replication, how does DNA replication occurs. Then transcription. Now let's know about the transcription. In this previous I have made a video on the expression of the genes which are involved in the protein in E. coli. You can visit and watch that video. In that it is given about the RNA, the role of messenger RNA, and all important points are given there. Let's know what is a transcription. It is a process of copying genetic information from one strand of a DNA into the RNA. It is termed as a transcription. So it is very important topic because many MCQs come from this particular topic. So the principle of complementary govern the process of transcription, except the adenosine complement now from the base pair with uracil instead of thymine. So we all know that if there is RNA, so it will not consist thymine. Instead of that, there will be uracil at the place of thymine. So unlike in the process of replication, which one set the total DNA of an organism where it get duplicated. So in transcription, only one segment of DNA and only one strand is copied into the RNA. So the boundaries that would demarcate the region and the strand of the DNA that would be transcribed. So here both the strands are not copied during transcription. So first, both strands act as a template. They would code for a RNA molecule with different sequence. So remember that complementary does not mean identical. And in turn, if they code for a protein, the sequence of amino acid in a protein would be different. So hence one segment of DNA would code for two different proteins, and this would complicate the genetic information transfer machinery. So second, the two RNA molecule if produced simultaneously would be complementary to each other, hence would form a double strand RNA, and this would prevent RNA from being translated into protein, and the exercise of transcription would become a futile one. So transcription have three units. The promoter, here we can see in the diagram there is a transcription starting site, and structural gene, a template strand, coding strand, and a terminator. So here the main three regions are promoter, structural gene, and terminator. So there is a convention in the definition that two strands of DNA and structure gene of a transcription unit, since the two strands have opposite polarity. DNA dependent RNA polymerase also catalyze the polymerization only in one direction, that is from 5' to 3', and this is the polarity of a 3' to 5' act as a template, and it is also known as the template strand. The other strand which has a polarity from 5' to 3' and the sequence same as RNA except thymine at a place of uracil is displaced during the transcription. Strangely, this strand which does not code for anything, it is referred as a coding strand. Now you must know what is a template strand and what is a coding strand. So here from 3' to 5' it is a template strand, and from 5' to 3' it is a coding strand. So here we can remember the sequence. We don't have to remember the sequence, only wherever there is A, the complementary will be T, and wherever there is G, the complementary will be C. This is important. Wherever there is 3 from 3 to 5 template, from 5 to 3 it is a coding strand. Then there are promoter, terminator, and structural gene in a transcription unit. The promoter is said to located towards 5' upstreaming of a structural gene. This reference is made with respect to the polarity of coding strand, and it is a DNA sequence that provide binding site of the RNA polymerase, and it is present on the promoter in transcription unit that also define the template and coding strand. So by switching its position with terminator, the definition of the coding and template strand could be reversed. So the terminator is located toward 3' end. The terminator is always on the downstreaming, and at the 5' end there is a promoter region. Here we can see in the diagram. So the terminator is located toward the 3' downstreaming of the coding strand, and it is usually defined as the end in the end of the process of transcription. So there are additionally regulatory sequence that may be present further upstream or downstream to the promoter. So some of the properties of these sequence shall be discussed. The transcriptional unit and a gene. So a gene is defined as a functional unit of inheritance. So the gene are located on the DNA, and gene, it is termed in the DNA sequence. So DNA sequence coding for tRNA or rRNA molecule, it is also known as a gene. So what is a cistron? At a segment of DNA coding of a polypeptide, the structural gene in a transcription unit could be a monocystonic, mostly in eukaryotic, and polycystonic, mostly in bacteria or prokaryotic. The detailed video I have made, you can watch about the polycystonic gene and its role in proteins. So in eukaryotes, the monocystonic structural gene have interrupted coding sequence. The gene in eukaryote are split, and the coding sequence or expression sequence are defined as an exon. So exon are said to be those sequence that appear in mature or a process RNA. So exon are interrupted by intron. So intron are intertwining sequences that do not appear in mature or processed RNA. So the split gene arrangement further complicate the definition of gene in terms of DNA segment. So inheritance of a character is also affected by the promoter and regulatory sequence of the structural gene. So sometimes the regulatory sequence are lost, defined as a regulatory gene, even though these sequence not code for RNA or a protein. So there are various types of RNA and their role in the process of transcription. So in bacteria, there are three major types of RNA, that are messenger, transfer, and ribosomal. So all these three RNA, they are important for the synthesis of protein in a cell. So mRNA, they provide the template. tRNA bring amino acid and read the genetic code. That is, the messenger RNA's work is to provide the template. The transfer RNA is what transferring amino acid, transferring and reading its genetic code, and rRNA, that is the ribosomal RNA, it plays in the structure and catalytic role when the translation is going on. So there is a single DNA dependent RNA polymerase that catalyze the transcription of all type of RNA in bacteria. So RNA polymerase, it binds to the promoter and initiate the transcription. It uses nucleoside triphosphate as a substrate and polymerase in a template dependent fashion following the rule of complementary. In somehow also facilitate the opening of helical continuous elongation. Only a short stretch of RNA remain bounded to the enzyme. So only the polymerase reaches the terminator region and the RNA falls off. So all the RNA polymerase, it results in termination of transcription. Then how this RNA polymerase is able to catalyze all the three steps, which are initiation, elongation, and termination. So RNA polymerase is only capable of catalyzing the process of elongation when it is associated with initiation factor sigma and termination factor to initiate the termination of the transcription. So associated with this factor, it alters the specificity of RNA polymerase to either initiate or terminate. So in bacteria, mRNA does not require any processing to become active, and also transcription and translation takes place in the same compartment. So there is no separation of cytosol and nucleus in bacteria. Many times the translation can begin much before the mRNA is fully transcribed. So the transcription and translation can be coupled in bacteria. In eukaryotic, there are two additional complexities. These are at least 3 RNA polymerase in nucleus in addition to RNA polymerase found in the organelle. So there is a clear cut division of labor. So RNA 1 polymerase 1 transcribe rRNA, that is 28S, 18S and 5.8S, whereas RNA polymerase 3 is responsible for transcription of tRNA, 5S rRNA, and snRNA, that is small nuclear RNA, and RNA polymerase 2 transcribe precursor of mRNA, the heterogeneous nuclear RNA, hnRNA. So what is important here? The initiation factor, the termination factor. Psi and phi. Psi for initiation, termination phi. Then types of RNA polymerase. RNA polymerase will transcribe the rRNA, that is 28S, 18S, 5.8S. rRNA polymerase 3 is responsible for transcription, tRNA, snRNA, 5S rRNA. Then RNA polymerase 2 will transcribe the precursor of messenger RNA. They are the hnRNA. So here we can form a table and learn this. Which RNA polymerase will transcribe which one. Then this one is very important one. Then next paragraph, the second complexity is that the primary transcription contain both the exon and intron, and they are non-functional. So hence the subject is to process this called splicing. Now what is it? When the introns they are removed and exon are joined in a defined order. So hnRNA undergo additional process called capping and tailing. So what is a capping and what is a tailing? Capping is unusual nucleotide like methyl guanosine triphosphate. This is added to the 5' end of hnRNA. So and the tailing, this is capping. What is capping? We are adding methyl guanosine triphosphate, where we are adding at the 5' end, where in hnRNA. And tailing is adenylate residue. They are 200 to 300. They are added at the 3' end. Tailing happens at 3' end. Capping happens at 5' end. This is important. Capping means adding what? Methyl guanosine triphosphate, where at the 5'. So tailing 3', capping 5'. This is easy way to learn. Then tailing, adenyl residue 200, 300 are added to the 3' end in a template independent manner, and it is fully processed in hnRNA. Now after fully processing, it is called mRNA. So there is a it is transported out of the nucleus for translation. So significance of such complex is now beginning to understood. So splitting gene arrangement, which are responsible for an ancient feature of genome. So the presence of intron and the process of splicing represent the dominance of RNA world. So understanding the RNA dependent processing in living system has assumed more importance. Now let's know about the genetic code. So during the replication and transcription, a nucleic acid, it was copied from another nucleic acid. So this process is based on the complementary. So translation required transfer of genetic material from a polymer of nucleotide Y to synthesize a polymer of amino acid. So there is no complementary strand existing between the nucleotide and amino acid, nor could be it is theoretically drawn. So they exist in ample evidence through supporting the notion that change in nucleic acid, that is the genetic material which was responsible for change in amino acid in a protein, and this lead to the genetic code that could direct the sequence of amino acid during the synthesis of protein. So if determined, the biochemical nature of genetic material and the structure of DNA was very exciting. So the proposition of genetic codes were mostly challenging, and in very true sense it require various disciplinaries who argue, since there are only 4 bases and if they have the code for 20 amino acid, you can learn 20 amino acid structure. I have made a video on that. You will know in detail how the structure is formed, the non-polar and polar group of amino acid. Then the code should consist of a combination of base, and he suggested that the order of the code for all the 20 amino acid, the code should be made above 3 nucleotide. This was very bold proposition because a premature combination, that is 4 raised to 3, would generate 64 codons. So the providing proof that the codon was a triplet was more daunting task. So the chemical method developed by Hargobind Khurana was instrumental in synthesizing the RNA molecule, with defined as combination of base, that is homopolymer, and copolymer. So Marshall Nirenberg free system of protein synthesis finally help the code to be decoded. So here there was a polynucleotide phosphorylate enzyme, the Severo-Oka enzyme, was also helpful in polymerizing the RNA with different sequence in a template independent manner. So enzyme synthesized for RNA, it finally a checkerboard for genetic code was prepared. Now we will draw this checkerboard in an easy way. So always remember, when you are drawing the code, there is always one sequence is repeating, like UU, we will write it a double double, and then UCAG, all the base pairs name we will write, and it will form. It was easy way you can learn. So similarly, each alphabet you have to write, like you are repeating U 4 times, then repeating C 4 times, repeating A and G 4 times. Then similarly in horizontal way and in vertical way we have to write it like this. So UA, UG, UC and UU. In this way we can write it. It is the simplest way to form the code for various amino acid sequences, and also the names of the particular amino acid sequences, the first, second and third position, which are given. Then silent features of genetic code are as follow. The code is a triplet. 61 codon code for an amino acid and 3 codon do not code for any. So hence they function as a stop codon. So some amino acid are coded by more than one codon, hence the code is degraded. So codon is read in mRNA in a continuous fashion and there is no punctuations. So code is nearly universal. For example, for bacteria to human, triple U would code for phenylalanine. Some exceptional to this rule have been found in mitochondrial codon and in some protozoans. So AUG has a dual function. It codes for methionine and it also acts as an initiation codon. Then UAA, UAG, UGA are stop terminator codon. It follow the sequence of a nucleotide in mRNA and predict the sequence of amino acid codon by taking help of a checkerboard. So stay tuned for the next video. Thanks for watching. Do like, share and subscribe.